I guess I can't be here 24 hours. Yeah, right, I'm just wondering how we're going to manage this. It's just a little bit.
I'm going to be up the whole dive, so I can step in.
We can take turns. Sure. Okay.
Okay, co-pilot in position.
Okay. Yeah. Okay, I have yours. I appreciate that. Thank you. Yeah. Okay, it's really simple to offer. I couldn't get the lights off, but then I realized, like, Hercules lights are boring.
Okay, could you give me a track line or?
I'm not gonna worry too much about that. But then.
That's no problem. We're just headed northwest. No, I was just thinking of that. Levi and I were talking about that. Okay.
Can I watch you fly?
Make me nervous? Yeah, sure. Excellent. I like that we have lights.
Magic. So
I think we can move in that general direction. So uphill, start, yeah.
Okay, taking off.
I got auto heading still in.
Yeah, that's fine.
Okay.
So the idea.
See if I remember how to fly this vehicle.
No, I just have to learn. When you lateral, it vibrates the main camera pretty good.
Yeah.
Learn that.
All right, everybody tuned in on nautiluslive.org. Thank you so much for joining us today as we are conducting our very first dive of NA174. My name is Brittany. I am the lead science communication fellow aboard EV Nautilus on this expedition.
Okay.
We just concluded our watch change, and so now it is the 4 to 8 crew. So once again, this is Brittany lead science communication fellow. Super duper excited to be back on EV Nautilus and to be back in this control room. And once again, thank you so much for joining us.
Auto.
Hmm?
You just made it auto. You wanted that. Okay.
Yeah, I'm good without having it in the auto.
Well, the no lights on mean it is auto.
That is auto.
Yeah.
Okay.
Is that what you want?
Yeah.
Okay.
There. Okay. Thank you.
See if we can get over this giant boulder.
So we basically want to continue moving uphill and look for rocks, potential rocks that we could collect.
Okay, roger.
And then you're pretty stretched out at the moment right now.
Yeah, I see that. You want to move the start moving the ship towards me? Okay.
So if you are just now joining us on nautiluslive.org, this is once again our first dive of this expedition, NA174, and we are currently exploring an unnamed seamount in the waters of the Marshall Islands. So our launch depth is about 2,200 meters, and we are expecting for this dive to go on for approximately 12 hours. As we're exploring, we are definitely looking for things that are of biological as well as geological importance. So we're keeping an eye out for anything that looks really interesting down here. And like I said, we did just have a watch change, and so we'll go ahead and do a round of introductions here in just a moment, but we're still getting settled here as we're making that shift.
Yeah.
Thank you, thank you. Danny, are you fine with me turning off your butt light there?
Yeah, go ahead.
Okay. There it is.
Wait for Atalanta to start swinging.
It's taking its sweet merry time.
Yeah, you can go ahead and zoom in.
We got a creature.
Biology.
Zooming. I know, I didn't mean to do it like this. Yeah. Yeah.
Sea cucumber.
Uh-huh.
Oh, yeah.
Danny, can you repeat your call?
Yeah, it looks like another type of sea cucumber. Yeah.
Yeah, that one definitely seems like it's more red It has more pigment than the other ones that we saw just a few minutes ago.
It's more red. It doesn't seem to have these like swimming appendages that the other one had. And he
Danny,
has those
are we
kind of
good
like
for an additional
spikes
zoom on video?
as well on his
Yeah, go ahead,
back.
zoom.
Zooming. So I do have somebody in the chat who's asking, what is the distance between the green laser dots? Yes.
Sorry.
Go for it, Daddy. Go for it.
The 10 centimeters.
Yeah, so those green laser dots are for measurements. They are 10 centimeters apart. So that sea cucumber looks like it's pretty close to 10 centimeters.
Yeah.
Almost perfectly.
And kind of bop up.
Oh my gosh, so cool.
Yeah, those lasers are really important to us because they allow us to be able to size the animals without having to collect them. It also allows us to kind of estimate the surface area that we see in the field of view, which we use for any type of quantitative analysis to estimate densities, for instance, of animals. This is really important to have these lasers on.
Good.
Thank you, Fanny.
Yeah, no problem.
That piece was good for the zoom. And then the way that I often will hold it is I got my hand, I'm doing like all three at once.
Okay, okay, cool.
I need to use this because you're helping.
Yeah.
We have something in the sediment too, if you see the little red stick.
Is that something you want to look at?
Yeah, can we zoom in on this, please?
Yeah, just a second, let me get a...
Right now you have SBL, so it's okay? Yeah. And always wait for the pilot to call for it.
Okay, go ahead and zoom.
Zooming?
And then about halfway.
Yes, that looks like a sea pen or a soft coral. Wow. So unlike the other corals that we might see during these expeditions, these are actually found in the sediment. And some of them, if you actually try to collect them, they might withdraw inside the sediment as well. So you can see here sort of what we call the polyps. So that's a colony of animals, and each polyp is an individual. And so that is a type of soft coral that we find in those soft sediments.
Yes, better.
Okay. That's amazing. So you're saying if we were to touch it, it would like retract very quickly into the sand?
Yes.
Oh, that's amazing. I didn't know that they could move so quickly.
Focus towards you. There you go. I know it's hard when it's similar colors, but this is helpful.
Actually,
I think we have some other settings here if you want it to be like that black and white.
So you remember how to do that? It's easy.
Okay, the ship's starting to get ahead of us, so...
Ready to go wide on video?
Yeah, go ahead and go wide.
All right, going wide.
all right, we can move on if you're ready.
And when you're coming wide, you can go a little bit faster, but still keeping it smooth. And then just checking your vignette there. Perfect. How does that feel? Good. Okay, good.
so I think we're about settled into our watch now. As I mentioned a few minutes ago, my name is Brittany. I'm the lead science communication fellow on EV Nautilus for this expedition, NA174. And I am not in this control van all by myself. I do have some lovely people joining me on this watch, and I would love to get some introductions in if possible. Hayden, would you like to go ahead and hop on and introduce yourself? You could tell us what your role is on this ship. How did you get here? What are you excited about?
Yeah, sorry about that. I was
no problem.
Focused on some things. So hello, I'm Hayden Niles. I am the one of the ocean science interns on board. I'm originally from South Dakota, but I go to the University of Hawaii at Hilo for college. I'm a marine science major. I've applied a few times before to this ship, and this was the one time that I got lucky. So I'm very fortunate, very thankful to be here, and very, very excited.
Awesome, Hayden. Thank you very much. And we do have a request in the chat. Somebody is asking if you could please let us know what your favorite deep sea creature is. I think I have an idea, but you can go ahead and say it.
Yeah. I'm a big fan of squid.
Yeah.
I like squids quite a bit. I could talk your ear off about them, but the one in particular that I like the most, I think, is the colossal squid. I think it's very interesting because of how mysterious it is, but I probably should not go into that.
Maybe save some for later.
Yes,
sure.
Of course.
Thanks, Hayden.
Thank you.
I see another critter.
Is there something you want to zoom in on?
No, I think we can move on. I think it's another sea cucumber here.
Yep.
We need to start a sea cucumber tally. It's like the 4th or 5th one we've seen, huh?
It's very common at this deeper depth and sedimented habitat because they are deposit feeders, so they basically feed on any detritus and material that deposit on the sediments, and so they usually like this type of environment.
I see. So you're saying because there's a lot of sand, that is what the sea cucumbers like, because they're basically like filter feeding through the sand.
Deposit feeding.
Deposit feeding.
Yes.
Thank you, Fanny. Well, Fanny, since you're sitting right next to me, lucky you, do you want to go ahead and introduce yourself a bit and tell us more about your background?
Sure. So I am an assistant professor at the University of Hawaii, Manoa, and I'm one of the co-science leads here on these expeditions. I'm a biologist, work primarily on deep sea corals, actually, so I'm really excited to see what we're going to find during this expedition. And it is also not my first Nautilus expedition, but last time I was on Nautilus was 10 years ago, actually, so quite a while ago, so really happy to be back.
Welcome back. And so I'm guessing your favorite deep sea creature would be a coral, right?
Actually, no. I would say it's also a squid, and I really like the big fin squid. It's only found in the deep ocean, and it has these very long tentacles, which I really hope that we will see during this expedition.
It's called a big fin squid.
Yes.
That's a good choice.
I need to immediately Google that. Big. I have never heard of such a thing.
Climbing the mountain.
On we go.
So before we continue on with our introductions, I do have a question coming in. Does anything eat the sea cucumbers? I'm inclined to say yes, but Fanny, could you tell us more? Does anything predate on the sea cucumbers?
That is an excellent question, and I am not entirely sure, but I would assume that some fish might eat them. But not fully my expertise, but I would assume so. I see another type of sea pen, if you can see here in the middle of the screen. I think that's a different type.
You wanting a zoom?
Yes, please.
Okay, let me get situated here. Go ahead and zoom video.
Zooming?
It kind of, yeah. So that's a different type. You can see it looks different. The other one had the polyps all along its body, right? And that one only has at the very top. It kind of look like some of the sea pens that
I have a little more zoom, Danny, if you want.
Umbellula, I would say. But a different genus than the one that we saw earlier. Super cool.
It's so beautiful.
Yeah, you can really see all these polyps here. So you can see those polyps. They have this. They look like feathers a little bit, and these are suspension feeders. So they are feeding on any type of particles that are in the water, and use those tentacles to filter all these particles and then eat them.
Danny, I had my Firefox quit on me, so just a heads up.
Yep, and you said that this is a type of Umbellula sea pen?
Yes.
I love saying that. Umbellula.
All right, thanks, Danny.
Okay, go ahead, go wide.
All right, going wide.
Okay, back up and running.
Excellent.
Slight zoom. There you go.
Okay.
All right, so as we continue making our way up this unnamed seamount, this is the 4 to 8 crew, and we are super duper excited to be on here with all of you who are joining on Nautilus Live. Once again, my name is Brittany. We've heard from Fanny as well as Hayden, and we do have some more in our control van that we would love to hear from too. So maybe we could hear from a few of our friends in the front row. Does anybody want to go first up there?
I can take a shot.
Yes, go for it.
Hi everyone, my name is Isabella Monaco. I am on the far right side of the front row. I am in the navigator seat, and I work with Ocean Exploration Trust as a contractor. So I do mapping and navigation.
Awesome, Isabella. Thank you so much.
Thank you.
And do you happen to have a favorite deep sea creature?
I spent the last couple minutes trying to really think about this, and I don't have a favorite yet.
All right.
So there's room for me to find one.
Challenge accepted. Another sea cucumber. That one's much bigger, it looks like.
Yeah, that looks like another different type.
It's like a darker purple.
It's a pretty purple.
Can we zoom in on this one, please?
Yep, let me just get situated.
Yes.
Isabella?
Yes.
Can you zoom out on this so I can see?
Oh, sorry.
Thank you. Perfect, thank you.
Yeah.
Actually, it looks like the one we saw earlier. The other reddish one that we saw earlier that looks like to be the same.
Right. This one, I don't know, to me it looks a tiny bit bigger. I don't know.
It is definitely bigger if you look at the lasers, but the type of, I would say, the species or type of sea cucumber should be the same.
Go ahead and zoom video.
Zooming?
So squishy.
Yeah, it's probably double the size as the other one.
Wow, you can see like the protrusions on its back.
Gonna continue to zoom?
Super zoom.
You're fully zoomed.
Amazing.
Wow, that's really cool.
Can you back out a little bit?
Backing out.
There we go. Let me get the lasers in there. Are you happy with that, Manny?
Yes, thank you.
Okay, go wide.
Going wide.
Hayden, I'm going to turn off Multipuffer 0 and 1 and start 2 and 3. Can you record that? Okay, 2 and 3 are turning on right now. Thank you.
Okay, can we continue?
I'm gonna zoom just a little to get that vignette out.
Yeah, we can
All
continue.
Right. Thank you, Danny.
Going uphill.
So there was a very interesting question that came in. Who discovered this seamount and how are they named?
What do you think, Mike? 5?
Great question.
Yeah, we could do 5 o'clock. Yeah.
So we actually did some sonar mapping earlier today over the seamount to get a better idea of the underwater topography of this area before we did the ROV dive. So it is currently unnamed. After we finish this dive, we will take the information that we have collected from doing the survey, and we will provide that to the people of the Marshall Islands, and they will be able to take that information and do with it what they will, and they can come up with a name for this seamount if they would like.
Yeah, I'm starting to get hauled around a little bit.
Yeah, I'm trying to catch up.
Yep.
Stop and smelling the roses for a little too long.
Too many sea cucumbers to take a gander, take a look at.
Say that again?
There's a rock.
No, we did 0 and 1 turned off and 2 and 3 turned on. Okay.
Oh, there's another sea cucumber.
Oh my goodness.
There's something else just below that sea cucumber.
Yeah,
I don't know if you saw it.
The ship's getting ahead of me. I gotta keep up.
He's stretching a little bit far out to starboard.
Yeah.
All right, I'm getting called out for not introducing myself fully, but whoa, there's a really cool thing we need to check out here first.
Okay.
Wow.
Yeah, it looks like it.
Is the ship stopped?
Yeah.
Okay. Yeah. Have the ship stop.
Okay.
What are we seeing here?
So that looks like Crinoid. So it's related to sea stars and urchins.
Drop down a little bit and give you a little bit extra.
Oh, actually, it is a Brisingid star, which is also the same related but different.
I'll give you a little bit. I don't want to go too far down.
So I've seen videos of crinoids that look like they're kind of swimming through the water. Are Brisingids able to do that too, or no?
So the crinoids, some of them are attached to the seafloor like a stalk, and some of them are able to swim. They have this kind of look like feathers that allows them to swim. I do not think that the Brisingid stars are able to do that.
That is a very, very big Brisingid.
Are you talking to me? So pump 0 was in milliliters, 134,581.
Zooming?
Correct, that's milliliters. And pump 1 was 129,496.
Okay, go wide.
Going wide.
I'd love to look at this thing all day, but we gotta go.
129,496.
I'm gonna zoom in for the vignette. All right, thank you.
Looks like a little belly button in the middle.
Okay, taking off.
It's actually its body, and its little mouth is on the other side.
Good to know. So because it's a sea star, it doesn't have any teeth, correct?
No, they don't. Sea stars have different strategies to eat. Some of them are going to go on their prey and are actually able to get their digestive system out and digest from outside the animals that then they eat.
They digest externally.
Yeah.
Such a wild way to eat.
Got a big boulder coming up.
Yep, I see it.
Oh, that is a big boulder. Yeah, somebody was asking earlier about the rocks that we're seeing. Are these mostly volcanic, or what is the origin of these rocks? Do we know that yet? That's a great question. And so I think that's what our geologists on board are trying to figure out. You can see that a lot of these rocks are covered in what's called a ferromanganese crust. And what that tells us is that these rocks have been here for a very, very, very long time. So I believe that our main geologist on board is very interested in doing some small collections of some of the rocks that we see on our dives so that we can bring them on board the ship and do some further analysis so that we can get a better idea of the origin. Are these volcanic, or did they come from somewhere else? I heard earlier on the previous watch that it looks like there was some sort of, I guess you could say, like a rock slide that occurred here long, long ago. So there's a lot of stuff that we can learn from these rocks, but a lot of it is unknown as of yet.
So I got a nut, and these are actually volcanic in origin, expected to be volcanic in origin.
Very cool. So we know that much, and then hopefully we
Atlanta.
can hopefully we can learn a bit more as we get those rock samples.
Captain Obvious, but
So let's go ahead and take some time to do some more intros. I'd love to hear from our ROV team if you two don't mind. Mike, can you go first? Tell us about yourself.
Sure. My name is Mike Burns. I'm your Atalanta pilot for the 4 to 8 shift. I'm an independent contractor for OET, so I work on this vessel.
Can we have a
as
quick look at this? Sorry.
Others? Currently living in California at the moment, and then I'd say my favorite deep sea creature is probably going to be a 6-gill shark.
Nice. Somebody was asking in the chat a couple minutes ago if we see a lot of sharks. We see them occasionally at depth, right? But there actually are 2 oceanic white-tip sharks that are circling our ship right now.
Yeah, I was going to say there's 2 right at the stern when we were launching the vessels.
Couple of shark buddies.
Okay, go ahead and zoom.
So this one should be a crinoid.
Oh yeah, I could see a bit of a difference.
So you can see, yeah, so you have this sort of like, actually I don't know the name in English, it looks like little legs.
Can you say it in French? Say the French version.
Actually, I'm not even sure. Like, they probably have some scientific name that I cannot remember right now. But that's basically like on the seafloor right now. And these probably can swim away. And it's kind of arms, see, they look like feathers.
Yeah, so beautiful.
Yeah.
Oh, okay. So they can get like a measurement? Oh, okay, cool. That's great advice, thank you.
Okay, go wide video.
Going wide.
Well, I guess I'll introduce myself.
Yes, please, go for it.
My name is Danny Hinshaw. I'm an ROV pilot for OET. I'm an independent contractor. And I've been doing this a few years now, and I love every time I come out here. My favorite sea creature is going to have to be just octopus in general, all the different variations.
All right, so far we have 3 cephalopod lovers, 1 elasmobranch lover, 1 undecided. Let's see what else we got. Kelsey, whenever you get the chance, would you love to introduce yourself to the people online?
Yeah, there we go, got my sound. Yeah, so my name is Kelsey. I am the video engineer intern this year. I'm from Alaska, and I think my favorite deep sea creature is actually a sea cucumber. I'm a big fan.
Oh wow, so you're very happy right now.
I'm stoked, yeah. Yeah, there was a marine like aquarium near my house, and we always would go see the sea cucumbers. So, big fan.
Amazing. Well, I'm very happy for you.
Thank you.
You're welcome. Okay, so we've met just about almost everybody on our 4 to 8 crew, but we have 1 person left that I would love to introduce you all to. This is Keone. Keone, can you tell us about yourself?
Hi everyone, my name is Euplectellid. I work with the Marshall Islands Conservation Society as a reef monitor, and yeah, very excited to be here. This is my first trip, my first time.
Sorry, we have something here that I wanted to check. Sorry.
All good. My first time checking out deep sea creatures, so right now I'm just looking forward to learn and see what I can find over here. It's very exciting. I don't really have a favorite deep sea creature right now. Hopefully that'll be determined at the end of this expedition.
Okay. You and Isabella both.
So
sounds great.
That looks like a black coral. So if we zoom in.
Sure. How's that? Right.
Yeah, we can hear you loud and clear now. Thank you.
Do you guys want to take more steps?
Hold here for us now.
Okay.
Okay, video.
All right, zooming.
Wow.
So that should be a black coral from the genus Stauropathes. And the reason why these are black corals is because their skeleton is actually usually black. So the coral itself is not black, but sometimes you can see in transparency that its skeleton, as you can see here on the image, is actually black in color.
Yeah, the bottom there.
I can get a little closer. All right, there we go.
Nice.
So as we take a beautiful look at this black coral, I can give you some background about the ROVs that we are using. When we are doing these dives on NA174, we are using a 2 ROV system.
Going wide?
So you met our ROV pilots for this watch just a couple of minutes ago. And we have ROV Hercules as well as ROV Atalanta. So when we're seeing this really beautiful imagery, that is coming from the amazing cameras that are on ROV Hercules. And ROV stands for remotely operated vehicle. So that means there is nobody that is in the water, nobody is inside of the ROV. Our ROV pilots that are inside of the control van are the ones who are controlling the ROV from the ship. And if you're able to look at your satellite feed number 2, you are seeing the image of Hercules from our other
Before
ROV.
we move on, sorry,
go ahead.
will it be possible to get a small piece of that coral?
Yeah, we could do that. Let me get back down to a place I can.
So in addition to having some really amazing cameras, our ROV Hercules also does have robotic arms. So it is able to use those arms in order to manipulate different things, move certain items, take small samples. So that's exactly what we're going to be doing here with this black coral.
I'll get it.
Mm-hmm. Oh, cool. That's so fun.
So it's a relatively small colony, so we don't want to take too much, but maybe like few branches in the, maybe somewhere in the middle.
Can you zoom in, video?
Yeah, zooming.
So just tell me what you are wanting.
So like a cut around here, maybe? Oops.
Around the middle and the top?
Yeah, middle top. We don't want to take too much, maybe somewhere over here.
Do you want us to zoom in a little more, Danny?
Yeah, go ahead.
Okay, I'm gonna zoom a little more.
Like that?
Let's see. Maybe a little bit more up, if you can. Yeah.
Okay, come back
I'm
wider
going to
so
come
you
back
can see
wider.
it.
Okay, where do you want it?
So these float away quite a bit, so
It's a dance.
maybe the forward.
Okay.
Do you want it in this section or?
We're gonna be pulling out the toolbox. Go ahead and pull the toolbox out.
Forward box or forward box coming open when you're ready.
Go ahead.
Okay, coming out.
Yeah, no IRs for this area.
Open it?
Yeah, open this bit.
Closing. Okay, closed.
Perfect, thank you.
And taking your dive again.
Nice, Danny.
Yeah, that was amazing.
Sometimes I surprise myself.
You don't surprise me.
Okay, are we good to go on?
Yes, ready
Okay.
to go.
Okay.
You can go ahead and get the ship moving. Thank you, Mike. That was excellent.
Absolutely.
Oh my goodness, another really, really big. I'm assuming that's a Brisingid, another one? That's even bigger than the first one.
Correct, and it looks like he has some arms growing. So what happened with sea stars, brittle stars, and Brisingid, if they lose an arm, it can grow back. So that can be a sign of cropping of a predator that might have eaten that Brisingid.
Okay, go ahead, video.
All right, zooming. All right, gonna go a little more.
Okay, I'm losing it. Go wide.
Going wide.
Okay. Atlanta's up ahead of me now.
I think that's just a bad DVO, but I could do a DVO reset.
Yeah, go ahead. That's better. I was like, how did Atlanta get over there?
Yeah.
Oh, yeah.
You'd be getting dragged.
You know what I found that I brought?
Right, so we have somebody who's curious to know about the sample that we just took of that black coral just a few minutes ago. When we do these types of sampling, what are we hoping to do with those samples? Do you have an idea, Fanny?
Yes, so mostly we're right now interested in the biodiversity in those environments. So these are areas that haven't not been explored before. And so basically we need to collect these samples so we can identify the species, because there's just so much that you can do from video. You can try to identify to some extent some of the species, but the best way to actually be sure about a species is to look at their genes and try to identify what they are. And so that's why we're collecting these samples so that we can analyze them and check which species they are to evaluate biodiversity in this area.
Is there something you guys? Is this anything you want to look at?
Maybe just to look what, I think this is just a dead stock from a coral probably, but there's white stuff on it. I'm not sure if that's anything. So you can zoom in on that.
Okay, let me get into a position. Okay, go ahead.
Zooming.
No.
No? Okay. Go wide.
Going wide.
So Fanny, was that a dead stock from a coral or a sponge? What are your ideas about that one?
I would say a coral because of the base and the way that it looks like. I mean, it's only a piece of it, but that would be my guess. I think we have a sponge in front of us.
Speaking of sponges.
Not sure it's alive though. We can zoom in on it, but
Go ahead, video.
Sorry, here you go.
yeah, that looked like one of these glass sponges.
Gonna zoom a little more.
But doesn't look alive.
Okay,
All
go
right.
wide.
Going wide. Okay, so non-vid live ESC time matches all time code windows. Captions. Oh, cool.
Out of curiosity, what about that sponge
Of the clothes.
makes you think it wasn't alive?
5248.
Mostly the color and part of it is gone, like a chunk of it.
Yeah, those look good. What's that? Proper audio.
All right, so we have quite a few questions coming in about our ROVs. We have some folks that are curious to know more about the cameras that are used. Are those cameras switched out pretty often? How often are they upgraded? And also, does it take a lot of patience in order to be able to maneuver the ROVs the way that you're doing, especially with the robotic arms?
The camera is on the vehicle. The main HD camera, the one the viewers are watching, is pretty old. It's the original camera to the vehicle. but it's still a very high quality camera.
We have
And then
something
we
else
have a
here.
wide field camera system, which was implemented a couple years ago. And that's given us our situational view and giving the pilot much.
Can we, sorry, we have like a few.
Yep, no problem. Stop the ship.
So on Hercules, the main camera that you're looking at is called a Zeus Plus camera, and that's given a lot of the really nice shots that you're seeing with the main screen. And on Atalanta, we have a Zeus Mini that is able to zoom in.
Oh, sorry.
Go ahead, video.
Zooming. Slow down, man. Zooming again.
Looks like another crinoid. Yeah. All right, we can continue.
Okay, go wide.
Zooming. Wide.
Okay. Do you want a picture? You want to look at that cucumber too? Or?
No.
Okay.
Sorry, Kelsey.
I'm only extremely disappointed. It's fine.
You got some to port. Hard to port.
Yep. Try to get away from it.
So this is a great time to reintroduce ourselves and reintroduce the dive to anybody that is joining us now who is just new to the stream. So my name is Brittany. I'm a lead science communication fellow aboard EV Nautilus, and we are currently exploring an unnamed seamount around the waters of the Marshall Islands. So we first started closer to about 2,200 meters, and we are making our way up the seamount. We're now at 2,114 meters. And the reason why we are exploring the seamount and why we are exploring these waters at all is because we're trying to get some baseline data. We are trying to get a better idea of what are the what's the biological makeup of this area, as well as the geological makeup of this region of the deep sea. And the more information we can gather about that, the better we can conserve this really amazing, precious area.
Fanny, I have a question. Did you want to keep heading in the direction that we're going, or did you want to follow more of the waypoint directions?
Sorry, so I want to continue going uphill towards that ridge. So the general, so you see what the waypoint 4 and then turning to waypoint 5. So we want to go up that ridge basically,
okay,
and go along it.
Okay.
There's some more sea pens here.
It's starting to get out ahead of me.
Yeah. Okay, go ahead and start the ship.
We're still going.
Oh, okay.
We'll take a couple more steps towards waypoint 4, but then we'll change bearings in a couple more steps.
Okay.
And as the ship's coming up, Danny, up right here to relieve.
Okay.
Okay.
Yeah.
So we have some people who are wondering if there are any larger animals that could be found down in these depths. Yeah, so from my experience being on this ship, I've seen lots of little ones, but I definitely do have some video highlights of some larger animals too, like some deep sea sharks.
Video's going to take a step.
We oftentimes find fossilized beaked whale bones in the deep sea. So there are different types of beaked whales that are really deep diving, sperm whales. Different types of octopuses and squids. I don't know if anybody on this ship or anybody on this watch has seen any of those larger creatures on any of their dives before. Sounds like not yet. But the expedition is young. We never know what we're going to find. Oh yeah, giant sponge. There was a really, really large sponge that we did find a couple of years ago when we were exploring around the Johnston Atoll region. That sponge was easily much larger than our ROV Hercules. How could I forget that sponge? I don't know. But yeah, that was definitely a humongous organism.
So if we can, could we go down and try to box some rocks? Yeah.
There's a little shrimp. Definitely seems like as we're making our way up the seamount, the rocks are becoming a bit more uniform in shape, and also it seems like there's starting to be less and less sand. So I'm expecting we're probably going to see less and less sea cucumbers once again, so sorry, Kelsey. But we'll see what else we can find as we make our way up this unnamed seamount. Danny, maybe you could help me answer this question. Somebody is curious about why are the corals growing apart rather than in a community? Like they do seem like they're pretty spread out and kind of sparse.
So they're pretty sparse here, probably because this is not an optimal environment for them. But if we get to a different areas where you have like strong currents and a lot of food, you will see them in large aggregations. Sometimes can be a lot of individuals together. So that's just a mark that the environment here might not be as suitable for these corals to thrive. So
Control Van, this is Data Lounge. There was some stuff over on the right that looked kind of good, rock-wise.
yeah.
It looks like it's slowly moving too.
So we want to, I guess that was not the area. So we're looking to some of those rocks, not right here, to see if they are moving when we poke them. But let's see if there is a good, not really a good candidate in the field of view right now.
What do you think that yellow spot was?
I only saw it briefly, maybe a sponge. Oh, it's back, it's back.
So I think I heard you say that's a type of sea urchin?
That's a sea urchin.
It is so round. I've never seen a yellow sea urchin before.
There's actually, if you can check
the
on the right here, there's a stick. Oh, there's a bunch of them. Oh.
There was a collection request for that
Crinoid.
urchin, if you see another one.
There is some interest in that urchin, I think, to collect that we saw. Yeah, it would be great if you could go back to that urchin, see if we can collect it. So we can.
Hey there. So I'm going to let you know, this is Megan in the lounge, and that is an irregular urchin called Pylometacrinus. And this was collected, I think, in the Wake region by the Okeanos explorer.
Right, so while we're working on trying to get this urchin collected, it does sound like this has been collected, I think, just maybe once before. So it's really interesting that we were seeing this urchin here around the Marshall Islands. All right, so getting to some questions in the chat. Can we do dives if conditions on the surface are bad? So probably not. The process of lowering the ROVs into the ocean and retrieving those ROVs after a dive is a very, very delicate, it needs to be a very careful process. And if there are rough seas, that is not the ideal time to get those ROVs in and out of the water. So definitely not doing any of these dives if the weather conditions are not favorable.
So do you think this could go in the bio box, or is it difficult to open the drawer from here?
So I do see a question here that I absolutely love, and I'm actually going to wait until Keone is available to answer it. But I do see a question about why we are studying this specific area, why the waters around the Marshall Islands. So yeah, I think it's going to be a very interesting answer, but I'll let Keone do that one. And I do see another question about, are we in a submarine type vessel? And the answer is no. So we are all on the ship EV Nautilus, and we are using ROVs to do these operations. So the video footage that you're seeing right now in the water on the ocean floor, that is coming from our ROV Hercules. And ROV stands for remotely operated vehicle. So there is nobody that is inside of the ROV. We are all on the ship, and we have ROV pilots who are here in the control van, and they are able to control those ROVs from the ship.
Yeah, before we sample it.
So we don't always have to zoom. We can ask them to zoom this way. We can ask them to zoom. So this has like, it's like 3 steps. So it'll be like in, medium, or out, basically. Oh, okay. But yeah, this is the pilot controller. And which camera is that? It's either sediment cam or ROV lower. Okay. Either this or the ship that we're in. Okay. Thanks all. You guys see nothing? Take a break for dinner. I paused the eating and said it was kind of seats.
So you might hear some different voices coming in and out of the control van. Within the next few minutes, it is actually our dinner time here on the ship. So we are having some of our team for our 4 to 8 crew being relieved so that they can go get their meals. And we are still focusing on this urchin, trying to figure out the best way to do that collecting. Seems to be a urchin that is very highly prized. Somebody is asking what's for dinner. I don't know. I haven't been down yet, but I can tell you the food on the ship is excellent. So I definitely eat better on the ship than I do at home. We get fed 3 meals a day, breakfast, lunch, and dinner, snacks, freshly baked goods, pretty much every single day too. The cookies are amazing. We had some oatmeal raisin cookies earlier today, which were probably the best I've ever had. So Steve, welcome back in the control room. It's great to be sitting next to you once again.
Yeah.
Yeah, it's been a couple of years.
Yeah.
There was a question that came in about sampling, and I was wondering if you could maybe provide an answer. Somebody was wondering about how do we select what to sample or not to sample, and is there ever a fear that we might be sampling something that could be potentially very rare?
The answer is we usually don't know. There's so few of these sampling events for a lot of these species. Even it's very common in the deep sea for two things to look similar, but to be distantly related. So these are, these are not exceptionally rare circumstances. They're rare on our dive track, but remember we're only sampling part of the seamount. But there's a whole population of things. There's a whole population of things on these sites, but for the most part, a lot of our sampling is directed by, potential for rarity, potential to support species descriptions, potential to support new range extensions, and if any of this. So some of the records we have for this irregular urchin are. Let's see how this goes first.
Yeah, of course. So somebody is wondering about this sea urchin that we are trying to collect. Just a few moments ago, you might have seen it kind of detach from the rock and start to roll on down to the location where it is now. And so, no, it wasn't running away. It was, so sea urchins, they have something called tube feet, which help it to pretty much hold on to any substrate. And so it probably just detached its tube feet and sucked those in, and then it was able to roll on down just because of gravity. So no, it wasn't necessarily running away, but
Danny,
we're still trying to figure out.
On that? Danny, do you want some zoom?
Do you want zoom, Mike, or are you good? So on these flush jar, so it shouldn't suck up, but if it does, it's going to go into that jar. Let me know when you're ready. Okay. Okay, you rotate your wrist. Okay. Maybe pull out a little bit more slack if you can. Okay. What's your grip force? Okay. Up, up. Okay. Moving on. Go ahead and put it back. Okay. Stow the arm. Okay. Yeah, it looks good. Okay. Okay.
All right.
All right, moving on.
Same direction? That's where I'm headed.
Maybe we'll find another one like
Zoom
that
in.
that we can collect.
Yep.
I can't hear you.
Oh no, no. If you touch it, it's now yours.
Hello, Multipuffer, turning on pumps 4 and 5. Did you get that?
Yep, copy that.
Thank
Pumps
you.
4 and 5.
And so is Herc kind of just moving like along the topographic gradient right now? Or is it going up or down in...
So we are
the...
actually moving up a ridge. So right now we're still not at the summit of that ridge yet.
Okay.
But the idea is to go up the ridge and then move along that ridge. And actually, if we could go upslope a little bit more, would that be possible? So we can, yeah.
Yep, we can go up the slope more.
Is that better?
Yes.
Okay.
Yes. Yeah, so pump 4 and 5 start, and end pump 2 and 3. It'd be sample 6 and sample 7.
You've got a float bounce up, Atalanta.
16 meters. You see it? Or, oh, yep.
We're seeing.
I got you.
I think
Thank
we've
you.
seen that.
I just
This
don't want to see it
Crinoid
wrap
earlier.
around.
Yep. Yeah. Okay.
Very
Should
cool.
give you nice and clear of it. Thank you.
Yep.
And so for those of you just joining, again, we are on our first dive of NA174.
Can you swap my camos back, please?
Back to dive salvo?
No. Swap my bucket cam back to my rail cam.
Oh, yep. I got you.
There we go.
You want port rail and starboard? Or do you want the starboard bio box?
That's fine.
Okay.
I can see around me.
So, Annette, when we are using this multipuffer to...
we have something here, I'm not sure.
Sorry?
No, that's might be just sediment, never mind.
Question?
Yeah, so when we're using this multipuffer to collect eDNA, is there anything specific that we're looking for?
Yeah, so we're taking
We have like another
samples
here
along a depth gradient.
On the right of the
that
screen,
we
if we could have a look at that.
Assess how biodiversity changes with depth as we move up the seamount.
Very cool. So we can kind of see how that environmental conditions may affect the biodiversity.
Right.
Amazing.
Annette, how many milliliters of water were filtered for 4 and 5?
For 4 and 5 are still ongoing.
Oh.
Okay. They were started
and zoom video?
So I have here coral, which may be a primnoid type of coral. So this is just a single branch with, and you can see the polyps here on the screen.
Sorry, Annette, I meant 2 and 3.
Okay. Pump 2, 117765. Pump 3, 113387. And those are in milliliters.
All right, thank you.
Yep.
Are you good?
Yes, we can go. Thank you.
Go wide.
Going wide?
So Fanny, those types of corals, if they're disturbed, will they retract?
Not these ones.
Okay.
So the one that we tried, the one that we saw in the sediment, right? So that was a sea pen.
Oh,
so these are a different type of corals. So they are called sometimes octocorals or alcyoniaceae. So they are generally, some of this one was not branched, but a lot of them are branched corals. So this type wasn't. So that coral cannot retract, but the polyps, if you saw those little polyps with their tentacles, so they can retract their tentacles.
Very interesting. Very cool. And they have little stinger cells?
They do. They're actually related to jellyfish. So they use these tentacles to catch food that is in suspension in the water column, and then they can eat it. So they are suspension feeders primarily. So corals are often found in areas where you are going to have strong currents. And obviously they need to be in areas that have enough food, which is possible that this particular area here is not fully suitable for these organisms to live in.
Yeah, go ahead and get the boat.
Gotcha. Very interesting.
All right, now go ahead and do your audio test on SPL. Without, like, talk without being on it. I'm talking to you on SPL currently now. Okay, I'll go talk.
Looks like there's another one of them.
Oh yeah.
Nav, can you get an audio check without going on SQL, please?
Can we have another look at it again, please?
Yep, no problem. Let me just settle down.
And we need to do a quick operational test for audio here. Just silent for everyone. Thank you. Everyone but Nav.
Go ahead and zoom video.
All right. Radio check, Nav. Let me do it without. Okay, I think we're okay. Thank you, Nav. Danny, do you want me to zoom a little more?
Can we focus a bit more on this, or is
I'm gonna zoom a little closer.
Yep, go ahead and zoom.
Actually, if we look at the skeleton, I was looking if you see these sort of black marks that could indicate that this is actually not a Primnoid coral, but a bamboo coral. If we see these what we call nodes, I don't see them here. The polyps also look more like bamboo coral. Interesting. Yeah, I think it's probably more, looks more like a bamboo coral.
Do you want to keep looking or go wide?
It might be worth collecting a small piece of it.
Okay.
And put it in the bio box with the black coral.
Okay, can you
go
zoom out
wide.
video?
Okay, I'm ready on the box when you are.
Okay, that was interesting. Oh, that's a problem.
Did craft just start to...
As soon as I pushed the blue button, it started falling down.
All right, so relatively small piece, maybe somewhere, oops, like here or a little bit, yeah.
Do you want a little zoom, Danny?
Hey guys, this is Lounge. Yeah, they said get the whole thing. Just talk to Steve. He was getting dinner. Sorry.
The whole thing?
Yep.
All right, so we want the whole thing.
Okay, at least half of it, Steve says.
Okay.
Okay, box out.
Okay, box coming out.
Video, you wide?
Fully wide.
You want me to
That's
retract
quite
back
long
in?
if those 2 dots are 10 centimeters.
I can retract in or see if that will do it.
Oh, bring the camera in?
Yeah.
I think we're there.
Yeah, we are. Okay. Closing. All right, we are closed.
Great, thank you so much.
Video's going to do a slight push for the vignette.
Okay.
All right, ready to go.
That was smooth work. Good.
We have 6 eDNA samples, just right. We're
Mike, can
collecting
you
4 and
make
5.
the craft come up? It's falling down again.
Yep. Got you.
We might have to just keep it on live.
So we have 6 eDNA plus 2 corals, so that sounds like 8.
Okay, coming on.
No problem.
You might just have to freeze it and leave the blue button on.
Yeah. Does that look good to you as far as the jaws and everything?
I'd close the jaws and lock them.
Okay. Okay, and you are frozen. Okay.
Okay. Sounds good.
Looks like we might have some kind of sponge in front of us.
Yeah, it looks like it's floating.
Yeah, it has a stalk.
So it's like swaying in the water.
There's something you want to look at?
Yes, please. You can look at that sponge.
Is there a reason that some of these have those long strands and not like one solid strand?
You mean the long stalk?
Like, see, it's just like a small little line. Why isn't it like one sturdy line instead?
Good question.
Just thought I'd
Go ahead,
ask.
video.
Yeah, if you can zoom in a little bit more, that would be great. Thank you.
Zooming.
Look at that.
Wow. It kind of looks like an eye, like with an optic nerve or something.
I can zoom a bit more.
But yeah, Hayden, I've seen sponges that have like a thicker stalk, and I just think that there's just different types of sponges.
I mean, sponges have a very high diversity of morphologies, I would say. In general, when we say in the deep sea, when there's something you don't really know what it often is a sponge.
Okay.
So they have like very diverse morphologies.
I love that. So maybe SpongeBob SquarePants really does exist down there somewhere.
Okay, go wide
That's
video.
a great comment. Yes, somewhere, right?
There is hope.
Yeah, that looks good, thank you.
Maybe if we go up to Bikini, we might find them.
Shrimp.
Maybe he's wandered down. Maybe he's getting close. Who knows?
I just went through. I accidentally skipped that. So.
Did you retract the camera or is it back out?
I did. I did not retract back when
Okay.
I touched it, but I will press right now.
No, we're good. I
Yeah.
just want to make sure I'm in the right.
Yep.
Is the ship moving? Okay. Yep.
So once again, we want to thank all of the viewers who are currently tuned in live online. We are about 4 hours into our dive. This is our very first dive of NA174. We are exploring an unnamed seamount in the waters of the Marshall Islands.
Copy.
So our primary objectives while we're doing these dives are we're trying to get a better idea of the diversity and distribution of deep sea animals, what kinds of living things are down in these depths. Specifically, we're looking at coral communities as well as sponge habitats, looking for what kinds of fish live down here. And we're trying to see if we can get to some baseline data for conservation value.
Can you porch out for me,
Porch out?
Mike? Yeah.
Yeah.
I think it's the same.
Porching out.
And the sponge. Some kind of hieronyma sponge.
Good.
And then in addition to
Oh, there's like a little...
Oops.
Looks like a little squat lobster on it. Wait, no. What is this?
Okay, video.
All right, gonna zoom.
Yeah, I guess it is.
The ship can keep moving. We're not going to get behind.
So that's the same coral that we just collected. And it seems that there is a small squat lobster on it.
Can you zoom out a little bit?
Zooming out.
There it is.
Do you see the Scott lobster?
I think move it down the camera a bit. Yes, there it is.
Good eye.
Steve requests that we collect the squat lobster, please. Suction.
Yeah, we can. Can we suction that lobster?
Yeah, we can.
Thanks, Penny.
Do you know what those long appendages are?
Yeah,
are
I
they
can do
arms?
it. Oh, you want to do it?
Yeah,
Go ahead.
Give it another whirl.
Can you see okay with the other cameras?
Yeah. Should be able to. I'm gonna actually.
It's amazing. It looks, at least on the camera, it looks like it's almost the exact same color as the coral.
I need to turn on my
Yeah, they
bucket
blend in
cam.
pretty well.
Yeah.